Cells are the fundamental building blocks of organisms. By that we mean that they are the simplest structure which is then either developed or divided into form many more new cells. Uh, this particular image is a nice chart which compares the relative sizes of living things and their components. So you can see, if you notice over here, the sizes of the cell are 10 to 100 microns, and if you compare that to a human, which is about 1 meter in length, or a blue whale, which is much larger, 10 times larger than a human, and compare the sizes of small structures such as chloroplasts, which are organelles present within the cell, or a primitive structure of viruses. Even bacteria are smaller than plant and animal cells, and the macromolecules that we talked about earlier are even smaller than the simplest viruses, and the atom is the smallest relative size that is shown over here. So you can also notice over here that the human eye can actually differentiate from from this size, about 250 microns to larger up to larger than a kilometer in size. However, the light microscope has a very narrow focus, and the light microscope usually will show you structures, but not in much clarity. You will see the cells visible with them. These are the light microscope are the ones that we will use in class during lab time, and you will be able to see some of the outlines of a cell and sometimes some larger organelles that are present. However, the book has some beautiful pictures, and all the images that I show you over here are usually taken by electron microscopes, which have a much higher resolution and clarity than a light microscope. The discovery of cell was actually done by Robert Hooke. Robert Hooke devised a very simple microscope, and he first looked at the structure of a cork, and that reminded him as a honeycomb structure. And because of a regular pattern in this honeycomb pattern of the cell, he named it as boxed structures or cell. Much later, when Lewin Hooke, who was another scientist, actually I'm going to write you these names. Robert Hooke, he was the one who looked at the cork cells, and later Antony von Leeuwen Hooke was the first biologist who actually observed living organisms in pond water, and he saw that there were things that are floating, and he then looked at more living matter than compared to Robert Hooke, who had looked at the cork cell by itself. Later, in the early 1800s, Robert Brown described one of the structures that was present in the cell as the nucleus. He was able to see the organelle much more clarity. After that, there were some German biologists, Sheldon and Schwann, and they also observed cells of various animals. And the study of cell is called as cytology. Cyto means cell, and cytology would be the study of the cell. And right now, the discipline of cytology is very, very large. The concept of the cell is based on a very simple primitive theory which is called as the cell theory. Cell theory was proposed by a German physician named Virchow. He proposed some postulates in this theory which were later proved, and we now all agree on it. His first statement was that all organisms are composed of cells. In other words, you have to have some sort of a structure in a living organism, whether it's a simple organism such as amoeba or a bacteria or a much complex substance like a person or a frog or a rabbit, or a plant or a tree. So all organisms, if they were alive, they were made up of cells. Cells are the simplest unit of living matter. In other words, if a cell is destroyed, if the bacteria is broken down into even simpler structures, that is, if you break the membrane apart and you open up the structures of the enclosed organism, then it does not stay alive anymore. His third statement was that all organisms come from pre-existing cell, which means that we can excuse me, just take out a magic wand and make organisms appear, or we can just disappear organisms. Cells will only arise from existing living cells. And that leads us to the next statement, which is cells are capable of self-reproduction, and they will produce new cells, whether it's the growth of an organism or whether it's for cell reproduction. So now that we know what a cell is, let's explore the other question, which is, why are cells so small? To answer this question, think of the surface area that is needed for a cell. If the cell is round, it needs to absorb as many nutrients and exchange waste for other substances, and thus constantly it will have things coming in and out of the cell. Now, if I took a small size and keep on expanding it and growing it, and if you really took out a measuring tape and measured the circumference around it compared to cell division, in which the cells would stay in the same volume, but noticed that they are of the same sizes, but if you took the surface to area and the volume of these small ones, you will find that this is a much larger volume. This is similar to the concept of cooking a steak or a pound of ground beef. Ground beef will cook quicker or faster because it has more surface area to volume ratio. So you can see in this image over here, in the same dimension that are shown over here, the volume of 64 1 centimeter cubes is giving you a total volume of the area is 384 centimeter square with the same volume. However, you can see the volume of the same object would be 96 centimeters square if it had just one big cube. So the main reason for the cells to be present in such a small size is because it increases the surface area of the cell. Now, some cells, for example, nerve cells and some large plant cells, are long and thin in order to keep the cytoplasm near the plasma membrane. There are other cells that are more specialized in absorption, and they will even have more surface area per volume of the cell, and this is done by having more invaginations or more folds on the plasma membrane or the outside membrane. Cell division will restore the amount of surface area needed for any exchange of these materials. So a cell will need a surface area that can adequately exchange materials with the environment. Surface area to volume considerations thus requires the cell to stay small and tiny.